Hello everyone! This is Sarah May Ligasa from Bachelor of Secondary Education, major in Science. And for today's video, I will be sharing my insights and learning from microbiology and parasitology, particularly from Unit 5 to 7. Unit 5, Microbial Metabolism. The things that we will talk about in Unit 5 are metabolism, enzymes, cellular respiration, fermentation, types of fermentation, and photosynthesis. Metabolism is the build-up and breakdown of nutrients within a cell. These chemical reactions provide energy and create substances that sustain life. The two key players in metabolism are: first, enzymes. It catalyzes reactions for specific molecules called substrates. Second is adenosine triphosphate, or ATP. It is a molecule that cells use to manage energy needs. If a reaction results in excess energy, some can be captured in the form of ATP's bands. Catabolic pathway results in the breakdown of more complex organic molecules into simpler substances. Catabolic reactions usually release energy, while on the other hand, anabolic pathway are simpler substances that are combined to form more complex molecules. Anabolic reactions usually require energy. Substances that can speed up a chemical reaction without being permanently altered in cells are called catalysts. In living cells, enzymes serve as biological catalysts. As catalysts, enzymes typically accelerate chemical reactions by lowering their activation energy. Factors influencing enzymatic activity. First is the temperature. At high temperatures, enzymes undergo denaturation and lose their catalytic properties. At low temperatures, the reaction rate decreases. Second is the pH. The pH at which enzymatic activity is maximal is known as the optimum pH. Third is the substrate concentration. Enzymatic activity increases as substrate concentration increases until the enzymes are saturated. And lastly, the inhibitors. Competitive inhibitors compete with the normal substrate for the active site of the enzyme. Non-competitive inhibitors act on other parts of the apoenzyme or on the cofactor and decrease the enzyme's ability to combine with the normal substrate. Cellular respiration has 3 steps, namely glycolysis, Krebs cycle, and electron transport chain. Fermentation releases energy from sugars or other organic molecules by oxidation. O2 or oxygen is not required in fermentation. Lactic acid fermentation. During glycolysis, which is the first phase of lactic acid fermentation, a molecule of glucose is oxidized to 2 molecules of pyruvic acid. Alcohol fermentation also begins with the glycolysis of a molecule of glucose to yield 2 molecules of pyruvic acid and 2 molecules of ATP. Alcohol fermentation is carried out by a number of bacteria and yeast. Photosynthesis is the process by which the energy of light is used in order to form energy-rich organic compounds from low-energy inorganic compounds. The sites of photosynthesis are mesophyll cells, stomata, chloroplast, stroma, thylakoid, grana, chlorophyll. The 2 stages of photosynthesis are light reaction, the photo part of photosynthesis, and the Calvin cycle that has 3 phases, namely carbon fixation, reduction, and regeneration of the CO2 acceptor.
Unit 6, Microbial Growth.
The things that we will talk about in Unit 6 are the requirements for growth, physical requirements, culture media, preserving bacterial cultures, phases of growth, and filtration. The growth of a population is an increase in the number of cells. The requirements for microbial growth are both physical and chemical. Physical requirements. Number 1 is the temperature. Most microorganisms grow well at the temperatures that humans favor. The minimum growth temperature is the lowest temperature at which a species will grow. Number 2 is the pH. Most bacteria grow best at a pH value between 6.5 and 7.5. Number 3 is the osmotic pressure. Microorganisms obtain almost all their nutrients in solution from the surrounding water. They require water for growth and their composition is 80 to 90% water. Chemical requirements. All organisms require a carbon source. Chemoheterotrophs use an organic molecule and autotrophs typically use carbon dioxide. Biofilms. Microbes adhere to surfaces and accumulate as biofilms on solid surfaces in contact with water. Most bacteria live in biofilms. Culture medium is a material prepared for the growth of bacteria in a laboratory. Microbes that grow and multiply in or on a culture medium are known as a culture. Chemically defined media is one in which the exact chemical composition is known. A complex medium is one in which the exact chemical composition varies slightly from batch to batch. Reducing media chemically remove molecule or oxygen that might interfere with the growth of anaerobes. Preserving bacterial cultures. Microbes can be preserved for long periods of time by deep freezing or lyophilization or freeze drying. The growth of bacterial cultures. First is the bacterial division. The normal reproductive method of bacteria is binary fission. Some bacteria reproduce by budding, aerial spore formation, and fragmentation. Second is the generation time. The time required for a cell to divide or a population to double is known as the generation time. And lastly, the logarithmic representation of bacterial populations. Bacterial division occurs according to a logarithmic progression: 2 cells, 4 cells, 8 cells, and so on. The phases of growth are log phase, stationary phase, and death phase. Infiltration. Bacteria are retained on the surface of membrane, filter, and then transferred to a culture medium to grow and subsequently be counted.
Unit 7 Microbial Control.
And lastly, for Unit 7, I will discuss about sterilization, actions of microbial control agents, pasteurization, filtration, types of disinfectants, and antibiotics. The control of microbial growth can prevent infections and food spoilage. One would think that canned food in the supermarket is completely sterile. In reality, the heat treatment required to ensure absolute sterility would unnecessarily degrade the quality of the food. Sterilization is the process of removing or destroying all microbial life on an object. Heating is the most commonly used. Sterilant, sterilizing agent. Commercial sterilization is heat treatment of canned goods to destroy C. botulinum in the spores. Disinfection is the process of reducing or inhibiting microbial growth on a non-living surface. Antisepsis is the process of reducing or inhibiting microorganisms on living tissue. Degerming is the swabbing of alcohol on the skin before receiving injection. Sanitation is to lower microbial counts to save public health levels and minimize the chances of disease transmission from one user to another. Alteration of membrane permeability. Certain chemical control agents damage the plasma membrane by altering its permeability, damage to proteins, and nucleic acids. Some microbial control agents damage cellular proteins by breaking hydrogen bonds and covalent bonds. Physical methods of microbial control are heat, moist heat, thermal death point or TDP, thermal death time or TDT, decimal reduction time or DRT, boiling 100%, autoclaving or steam under pressure. Pasteurization is a process named after scientist Louis Pasteur that is employed to eliminate pathogenic microbes on milk. Filtration is the passage of a liquid or gas through a filter with pores small enough to retain microbes. Microbes can be removed from air by high-efficiency particulate air filters. Antibiotics are medicines that help stop infections caused by bacteria. They do this by killing the bacteria or by keeping them from copying themselves or reproducing. Antibiotic resistance. Because antibiotic use is the primary driver of antibiotic resistance, it is important to know when an antibiotic is truly necessary. Microbial characteristics and microbial control. Gram-negative bacteria are generally more resistant than the gram-positive bacteria to disinfectants and antiseptics. Mycobacteria, endospores, and protozoan cysts and oocysts are very resistant to disinfectants and antiseptics. Non-enveloped viruses are generally more resistant than enveloped viruses to disinfectants and antiseptics. Prions are resistant to disinfection and autoclaving. To destroy prions, infected animal carcasses are incinerated. Thank you so much guys for watching and for listening. And once again, I am Sarah May Ligaso from Bachelor of Secondary Education, major in Science.